Okay, Dr. Hannah. Bismillahirrahmanirrahim. Assalamu alaikum and good day, ladies and gentlemen. I'm Hazima Mozam and I welcome you to Comstech Distinguished Scholars webinar series. We have an exciting lecture in store for you. This lecture will be delivered by Dr. Ikram Burney. Dr. Ikram Burney is a senior consultant in medical oncology at Sultan Qaboos University Hospital, Muscat, Oman. And he's also a visiting faculty at Panjwani Center for Molecular Medicine and Drug Research, International Center for Chemical and Biological Sciences, University of Karachi, Pakistan. The title of his talk today is In Search of Improving Outcomes Across the Continuum of Ovarian Cancer. And with this, I would request His Excellency, Professor Dr. Iqbal Chaudhry, Coordinator General Comstech and Director ICCBS, to kindly introduce and welcome our guest speaker for today. Over to you, sir.
Azima, thank you very much. Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh to all participants. I'm sure that many of them would be joining us very soon. One thing which I have often noted that good clinicians do not really come into research. And as a result of it, you will really find two different streams of healthcare. One in which basic scientists are working on understanding diseases at the molecular level with no understanding of clinical outcome. And on the other side, you'll find lots of clinicians with so much of rich experience of treating people with little understanding of the etiology at molecular level. This is exactly what is important and where Dr. Ikram Burney impressed me a lot. Dr. Ikram is a clinician par excellence and a researcher of a very high quality and extremely high caliber. He has absolutely phenomenal experience of working in the field of oncology, clinical practices, and then on top of it, he's actually working on the science of oncology, the basis of how to treat people. And this is where I find Dr. Ikram extremely relevant for all of you, all the participants who are looking for a researcher and a clinician who understand both aspects of it. Of course, I know Dr. Ikram since long. He is a professor at our center and very well known internationally and regionally for his contributions in the field of ovarian cancer. I know him personally and professionally. We were engaged in some research projects also, and we hope to have a more vigorous collaboration as we move forward and move out of this mobility constraint. But one thing is for sure that he is one of those individuals who wish to engage with people, both for developing their understanding and capacity and also to engage them in meaningful dialogues, teaching them the fundamental of a lot of very important things. I have seen Dr. Ikram's review of the students who attended his courses, and I was extremely impressed from the kind of input we have received, the feedback we have received from them, and it is the credit goes to Dr. Ikram. Dr. Ikram is a senior consultant in medical oncology at Sultan Qaboos University Hospital in Oman and a visiting faculty at our center, which is Dr. Panjwani Center for Molecular Medicine. He is graduated from the Liaquat Medical College, University of Sindh, and received his training in medicine and medical biology at Hammersmith Hospital, London, and Aga Khan University Hospital in Karachi. He also worked in Southampton University Hospital Trust in UK. He worked as a consultant oncology at Aga Khan University, which is known for his excellence. Since 2003, he has been working at Sultan Qaboos University Hospital as a senior consultant in medical oncology. Areas of his clinical and healthcare research interests include lymphoproliferative disorders, gynecological malignancy, sarcoma, supportive care and oncology, and more recently, medical education, where I find His excellence appears in many ways and benefit lots of people. He chairs the curriculum committee at the College of Medicine and Health Sciences at Sultan Qaboos. I will not introduce the abstract, it's also already provided to you, but the topic is extremely important because we all understand that breast cancer and ovarian cancers are the major pillars of people all around the world. Over to you, sir. Ikram Burney, sir.
Thank you very much, Your Excellency, for this very, very kind and generous introduction. I would like to thank Your Excellency, Professor Iqbal Chaudhry, for giving me this opportunity to give a talk here in this forum. It's indeed an honor and a privilege for me. I would also like to thank the organizers of the meet for their very kind management of the of my talk as well. What I would like to do today is to give you. Let me share my slides over here. Is to give you an idea of what we are doing here for the past few years in one field, which is ovarian cancer, which is one of the very important cancers that I treat and which has got a lot of important recognition in the recent years. And I will basically introduce to you some ongoing work that we are doing and then look forward to collaborations as well. And here are my relevant disclosures. And the agenda is here. I'll talk very briefly about the burden of disease, talk about the importance of the cancer continuum. And I think that is very important when we talk about any sort of a research in any area. We think about disease not in its current form or in its current position, but is over the continuum as well, and see where interventions are possible and can be done and are required. I would then share with you some data about the work that we have been doing, our group, on the early detection of ovarian cancer, and then some data on the overcoming the resistance to the most common and the most important drug, which is cisplatin, and then I'll obviously conclude. Ovarian cancer is not the most common cancer, as you can see from the Globocan data. It's the breast, colorectal, and lung cancer, which occupy the almost 1/3 or half of all the cancers. Nevertheless, ovarian cancer is more than 3% of all cancers. As you can see on the left-hand side of the screen with this arrow, 3.4% of all cancers are ovarian cancers across the world. But more importantly, when you look at the mortality on the right-hand side of the screen, you see that 4.4% of all cancer deaths are related to ovarian cancer. Now, what does that mean? Which means that people who are diagnosed to have cancer are treated of their cancer, they get cured of their cancer, and they live and die because of other reasons. But here in ovarian cancer, people continue to die because of the disease. And this is a major and a huge issue with ovarian cancer. Just plug the battery here as well. Yeah. However, it's important to then also know that ovarian cancer is not one disease. We talk about the epithelial ovarian cancer, which constitutes 70 to 80% of all ovarian cancers. And then the less common forms, which are also called ovarian cancers to a common person, such as the germ cell cancer, sertoli-Leydig, and granulosa theca cell tumors. But those are very, very curable cancers, and some of them can be cured only with surgical resection as well. Even within the epithelial ovarian cancer, things are not as simple as they may appear on the surface, because you have got these different types of cancers, which have got obviously subtypes as well, different pathology, and the good pathologist would be able to actually diagnose them on H&E, and sometimes using the more new immunohistochemical markers as well. More importantly, there is a huge difference between the molecular mechanisms which govern the etiology of these cancers. For example, the high-grade epithelial ovarian cancer compared to the low-grade mucinous and etc., so on and so forth. And then some of them are sensitive initially, at least to platinum-based chemotherapy, others are not. And even within the most common form, which is the serous papillary type, this is the heat map, which shows that not only you have tumors which are genetically very different, the high-grade tumors over here in the green, and the tumors of low malignant potentials, which are tumors which usually occur in patients are diagnosed in stage 1, and patients are actually cured of their cancer after surgical resection. And the pathways of disease are different. The MAP kinase pathway governs the, by far and large, the tumors of low malignant potential, such as the tumors of the serous carcinomas of the ovary. And then you have got the other molecular mechanisms for the high-grade cancers, which are mainly p53 deleted. Now, for the purposes of talk today, I will obviously concentrate only on this very small one part, but which is the major part of ovarian cancer. And we will talk about the high-grade epithelial ovarian cancer. Although it is one type of ovarian cancer, but by far and large, about 70 to 80% of all ovarian cancers are high-grade epithelial ovarian cancers. And that is why the mortality the figure that I showed you initially is because of this high-grade epithelial ovarian cancer. What you see on this slide is that if patients were diagnosed in stage 1, which is disease confined to the ovary, then the cure rates are in order of around 80%, whereas if they were diagnosed in stage 3 and 4, we talk of cure rates of 5-year survival rates down to less than 20%. And that has not changed over the years. And I will show you some data on that as well. Now, with that, I would like to introduce you to, for those of you who might be interested in research in cancers, is that when you think of research in any kind of disease or any kind of care, usually for most of the physicians, clinicians, most of us, we talk about diagnosis, treatment. What we probably don't think a lot is the prevention, and also ways of early detection through screening. And those are very important, because if we were able to do that, then we would have an effect on the outcomes. And also something which is important afterwards, the treatment is now, and there is abundance of research which is coming out, is the issues of survivorship. What if the person lives beyond 5 years, but then they would have not only the psychological, physical, emotional, spiritual trauma as a result of the disease, and how do they cope with it? And those survivorship issues are also important to look at. And obviously, terminal supportive care is an area of work as well for those who are actively involved in the clinical management of ovarian cancer. I would not go into details of primary prevention and secondary prevention, but I would just introduce to you and say that primary prevention just does not work in ovarian cancer. There are no convincing evidence, except for what is known for all the cancers, which is leading a healthy lifestyle, which is eating right, doing exercise, and doing the right things. So those lifestyle modifications also apply for ovarian cancer, but nothing in particular. What does apply to ovarian cancer is the secondary prevention, because we know the definitive risk factor, and the risk factors is a family history, the most common of which is the hereditary breast and ovarian cancer syndrome, as you know, driven by the BRCA gene. And then there are less common ones, and we are not going into details of that. But this slide, what it tells you, that the lifetime risk of somebody getting an ovarian cancer who has got BRCA1 mutation is about 45% over there. And for a BRCA2 mutation is about 12%. So it follows then that if it were known that there was a family history, and if there were mutations, then obviously you need to do something about the ovary here and there in BRCA1 and BRCA2 mutations. And the treatment over there these days obviously is a risk-reducing salpingo oophorectomy, which is born out of evidence. It results in marked reduction in the incidence of cancers, and it's a recommendation now, a straight recommendation. However, the caveat here is that only 5 to 15% of all patients with ovarian cancer have got have mutations in BRCA1 or BRCA2 gene, which means that for the vast majority, for 85 to 90%, this sort of a secondary prevention is not an option. It became it came to headlines and to light when Angelina Jolie, way back in 2013, got bilateral salpingo oophorectomy followed by bilateral mastectomy because the family has got BRCA1 mutations, and as a result of that, the general public, what they did, they had a huge influx into the genetic centers asking for the mutation. The referral pattern increases, and as a result of that, there were more detection of BRCA1 and BRCA2 mutations in general practice as well. And here is the effect of the celebrities when they show something and then people follow. And just before the talk, myself and Professor Iqbal were talking about the issue of the uptake rate of vaccine. I think one of the ways of what media can do is to bring to fore the celebrities who are getting vaccine, and then that probably would lead the way for the public to do the same thing as well. Now, what I would like to do is not talk about prevention, diagnosis, or the rest of it. For the purposes of talk today, talk only about two areas that I have been working together with lots of my colleagues and collaborators, which is screening and early detection and treatment. So I move to the screening. Conventional screening through ultrasounds and a tumor marker called CA125 does not work. And as recent as May of this year, a couple of months back, there was this publication in the Lancet Oncology, which is the UK tox study, which had randomized patients into 3 arms: the no screening arm, the ultrasound, and the multimodality screening arm. And what they showed was that although you could detect, for example, here on the left-hand side of the screen, the low-grade and the high-grade tumors a little bit better as compared to the no screening, but ultimately there was no effect on mortality. And these are data from after 16 years of median follow-up time of 16 years. So clearly, the conventional methods of screening, ultrasound and multimodality do not work. Our group, we got interested in looking at the molecular markers or biomarkers to see if we can suggest markers which can help. We selected some cell lines, the MCAS, which is a mucinous adenocarcinoma, the cisplatin sensitive and resistant cell line, the A2780S, and this counterpart, which is resistant to cisplatin, CP, and the OFSAHO, which is a high grade. And we also use the HOS6, which is the normal ovarian normal cell line. And the first thing that obviously we did was the chip sequencing. So basically you take the DNA, you cross-link it, you immunoprecipitate it, and then you look at the genes and then run the PCR and see what are the genes which are involved in a particular tumor type. And the outcome analysis here showed that there were 36 genes upregulated in the MCAS, which is the low-grade ovarian cancer cell line, and 68 in the OFSAHO. But 14 genes were upregulated in both, which were common. And here is a rundown of some of those which were expressed. And we started working on one of those, the FAT4 gene. And I'll show you some data related to that, which is controlled by the Hippo signaling pathway. Now, it makes sense to think that not only if the gene is upregulated, then you look at the transcription factor upstream as well to see if you can control that as well. And the transcription factor upstream of the FAT4 gene and ERIT gene and other genes is over here, the E2F. E2F is involved in the early stages of epithelial ovarian cancer, and the downstream genes are affected by that. E2F leads to cell proliferation, cell cycle progression, and transformation as well. So we started looking into the expression of E2F. And here are some of the results. You see the messenger RNA levels low in MCAS, but high in the OFSAHO cell line compared to the normal ovarian cells, and very high in cisplatin sensitive and resistant cell lines, which are derived from the endometrioid type of of ovarian cancer. And here are the protein levels as well. Once again, you see that there is a increased expression of the proteins from the Ovsaho cell line. My colleagues and I then looked at the database, the gene expression profiling interactive analysis, and saw that it is not only here in the cell lines, but also if you look at about 426, on the database, there are about 426 ovarian cancers and about 88 or 90 normal tissue cell lines, normal tissues as well. When you look at the expression, you see that E2F indeed is upregulated in the vast majority of the tumors and only a small minority of the normal tissue as well. So the next question was to see what is the functional role of E25? Is it a bystander or does it do something as well? And for that, we use the silencing of we silence the RNA using the siRNA. And what you see is that indeed you can silence the E2F by using the siRNA, both in the form of in the MCAS and the Ovsaho cell lines. And once you do that, you then look at some of the assays over there, for example, the cell viability assay, and you see that patients, the lines which were treated, I'm sorry, I talk about patients because I talk about patients more, but here the cell lines, what you see is the E2F, the siRNA treated E2F cells, there was a reduced cell viability, there was a reduced colony forming unit both across the MCAS and the Ovsaho cell lines, and there was also a reduction in the invasive potential as well, as seen over here. And this was recently published, and then we've worked further to suggest that this actually is regulated by the Hippo and the Wnt pathways. I'll come back to that in a minute as well. Now, the genes which are involved downstream of E2F5, I spoke about the FAT4 gene, but there are other very important genes which have got now we know clinical significance as well, such as the ARID1A, and there are actually inhibitors, as some of you may know, of ARID1A inhibitors trying to treat the ovarian cancer, and then the FBXW7 gene. So these are important genes, and we have looked at one of those, which is the FAT4 gene, which we know also is upregulated or is a tumor suppressor gene. So loss of expression of FAT4 would lead to tumors of the stomach and the breast as well. It's a tumor suppressor gene, and it's, as I just mentioned, it's the downregulation is associated with pathogenesis of breast and gastric cancers, and there are lots of data on the screening, the mutational screening, which show that there may be mutations in many cancers, including the mucinous ovarian cancers as well. So here we looked at the serous ovarian cancer. So the first thing that obviously we did was to look at the FAT4 expression and its inhibition with the siRNA. And here we see that compared to the normal cell, the expression of the gene was indeed markedly low in the low grade as well as the high grade ovarian cancer cell lines, and so was the protein expression. And when you inhibit the genes with the siRNA, you saw that there was a further reduction in the so the tumor suppressor gene is, as you know, a molecular policeman. So suppression of that should then lead to upregulation of lots of the proliferative activity, and that's what we wanted to test by doing the siRNA, is that you could actually inhibit the gene using the siRNA for fat, and then looking at the FAT4, the functional role, what you see is that the cell viability is increased in the MCAS cell line as well as in the Ovsaho cell line. Then you see the soft agar over here. There is an increased colony forming over there. Then you see the colony forming using the, this is the crystal violet, I'm sorry, this is the soft agar over there. And then the cell invasion, which also increased as a result of the blocking of the FAT4 gene. So here is a evidence to suggest that FAT4 has got a role to play in the early ovarian carcinogenesis, and if identified, could be used as a potential marker. So the question then is that if you suppress the FAT4, then what proteins are the functional proteins which would be seen. And what you see here is some very nice data compared to the controls. And I think this is significant, because what you see is a reduction in the E-cadherin expression and an increase in the vimentin, suggesting an epithelial mesenchymal transition, or EMT, as we know. We also saw an increased expression of the YAP protein over here, which is the second arrow on the right. I'm sorry, I'm missing on the cursor here. The second protein on the right, the YAP protein, which is a part of the hippo pathway, as you know. And then a whole lots of proteins lower down, the cyclin D1, the CDK1, and BCL2, all involved in proliferation and apoptosis. On the left-hand side over here, what you see is the unphosphorylated portion of the YAP, suggesting that this is indeed the process of the process of expression of proteins is governed by the suppression of the FAT4 gene through the hippo pathway. So based on all of that, we we have hypothesized that over here the FAT4 repression of this leads to upregulation of YAP, which is part of the hippo pathway, and beta-catenin, which is part of the Wnt beta-catenin pathway, and those lead to then mesenchymal proliferation and markers of proliferation, and then that leads to metastases, etc. So if you were to diagnose or detect FAT4 early on in the stage of carcinogenesis, that might serve as a potential marker, and obviously this requires a lot of further work. What we also saw was that there was an inverse relationship between the FAT4 and the E2F5. So what you see over here is that if the FAT4 expression increases, the transcription factor goes down. So there is an inverse relationship over here, which is also something which is very interesting and exciting. I now would like to change gears and move on to the other part, which is the treatment of high-grade epithelial ovarian cancer, and would like to introduce to you what me as a clinician has seen over the last 25 years in the clinical practice, and more recently what I have seen. What I have seen over the last several years is that if you use that combination or that combination, or a combination of three, or a sequential, 5 different chemotherapy regimens, and the long-term outcomes of high-grade epithelial ovarian cancer is virtually the same. The median survival, median progression-free survival, something in the region of about 15 months or so, and only about 10 to 15% of people crossing the barrier of 5 years over here. And then also the overall survival, which sort of never exceeded about 48 months or 4 years, no matter how hard you try with conventional cytotoxic chemotherapy. So clearly, this data came out in 2005, and after that, there was no further attempts to use more or chemotherapy to try and treat the ovarian cancer. In the last few years, in the last, let's say 8 to 9 years, since 2012, when we learned about the fact that inhibition of PARP may work, the PARP enzyme, which is, you've got one allele which is inactivated as a result of BRCA mutation, and when the mechanism of repair is the base excision repair, and if you inhibit the enzyme which causes base excision repair, which are the PARP inhibitors, then you can achieve better control over the tumors. And obviously there are about 4 or 5 drugs in the market. The first one which to come out was this drug called olaparib. And what people have shown is that the progression-free survival, for example, would increase from about 5 months to 30 months. Now that is huge. That is very good news for lots of patients with ovarian cancer. But as I said initially, only 5 to 15% of patients have got BRCA mutations. Some other have got BRCA-ness, which means that they've got other genes which can cause this sort of a state of a sensitivity to PARP inhibitors, but not all. And furthermore, the natural history of ovarian cancer tells us something which is not so very pleasing. The natural history is that symptoms go on for about 6 to 8 months. before we diagnose ovarian cancer. Once the ovarian cancer is diagnosed, we treat them with surgery and chemotherapy, or with maintenance chemotherapy or maintenance PARP inhibitors, as I just showed you. But the progression-free survival is in the region of about 12 to 30 months. And then most of the patients would recur. Almost 80% of the patients would recur. 60 to 80% of patients would recur. And then they would be treated with a second line and third line and a fourth line chemotherapy. And then there is not a very good chance of survival after that. And here are some of the most recent trials. And I think what I would like you to concentrate here on this very busy slide is these lines over here in the blue color, the progression-free survival, which is despite the two trials on the left side, the desktop 3 and the GOG 3213, which looked at the surgical resection of the residual ovarian cancer, and the SOLO 2 and SERI 9, which looked at the use of PARP inhibitor in ovarian cancer, the progression-free survival does not go beyond 20 months. And the overall survival, as you can see, it does not go beyond 4 to 5 years. The maximum that you could probably achieve over here is 51 to 53 months, which is 4.5 years. So things are not all that very rosy over there. And once again, the issue is that it is a law of diminishing returns. Once the recurrence occurs, you can treat. The second recurrence occurs, you can treat further. A third recurrence occurs. The time it takes for the subsequent lines of chemotherapy gets shorter and shorter, called the progression-free survival. And what you see is that after about second recurrence, it goes down from 6 months to 5 months to 4 months, even if you treat with multiple lines of chemotherapy. So clearly there is a huge unmet need to identify drugs which could be used at that stage. And the two most important drugs in ovarian cancer are cisplatin and paclitaxel. And most of the patients who would ultimately go on to die because of ovarian cancer would have developed resistance to both the cisplatin and paclitaxel as well. And it is at that stage where we sort of tried to look into ways of overcoming this resistance. So going back to the drawing board, we selected those two cell lines, cisplatin sensitive and resistant, called the A2780S and the A2780CP. Now this cell line was derived from a patient with high-grade epithelial ovarian cancer, and then run through 6 or 7 passages with low doses of cisplatin to induce resistance. And the resistant cell line is 6.5 times more resistant compared to the sensitive cell line. And we also use the immortalized ovarian cancer cell line, and we started using some of the compounds locally available in Oman. The first one of which is with one of my earlier PhD students, Kamla, now Dr. Kamla. She looked at the Omani frankincense, the acetyl ketoboswellic acid, and what she was able to demonstrate was that there is indeed a time and dose response curve using with increasing doses of the ACPA, the O17, the acetyl ketoboswellic acid. The IC50 was in the region of about 20 to 21. And what we obviously did further was to look at the time of exposure and the dose, as you would usually do. And this was the dose which was selected around about at 25 micromole. And then the further experiments were run with that and looked at the protein expression, the protein expression, and found that there was indeed a high expression of pro-caspase 3 over here, and the cleaved caspase 3, suggesting that this was indeed mediated through apoptosis. And when we sort of looked at the through flow cytometry, which stage of apoptosis was seen more frequently, then at the increasing doses, what you see is that it's over here, the early apoptosis in the red over here, the late apoptosis in the green, and the debris. So there was some degree of necrosis, but mainly it was a late and early apoptosis, and the viable cells, at least at that concentration, were very, very few. Around the same time, sometime in 2015 or so, we had a lucky break. One of my collaborators over here, Dr. Sergei Dobretsov, who worked in the college next door to us in College of Agriculture and Marine Sciences, came over and said the usual thing, you know. He said, I dive into the sea next to the coast of Oman over here. This is Muscat over here. So he dives somewhere over there in those two areas, Bandar Rauda and Bandar Al Khairan, and he isolated about 40 different compounds from there, which included sponges and algae and corals and tunicates and etc. And he came over to me and my other colleague, Dr. Yahya Tamimi, and said that can we do something about them? And we said, okay, let's have a look. And this is something which we published. And I had the privilege of presenting some of this work at the ICCBS meeting a few years back as well. And once again, it was back to the basics. So we used MCF7 cell line over here, which is, as you know, a low-grade breast cancer cell line. And these marine organisms, which were collected from there, were extracted. They were separated. You know these things better than I do. And then compounds were identified using the NMR. Sergey did that for us. And then we started dropping the compounds on the cells. And indeed, what we saw obviously were that it induces apoptosis. And here is the electron microscopy showing that it induces the cytoplasmic blebs and signs of necrosis and apoptosis. And that was great. And we said, okay, let's look at all of those. We published that. And what we found was that there was indeed the anti-cytotoxic activity, which was medium to strong to weak to medium to strong. And we identified compounds like hibernality, melformin A1, the economine, Gallic acid, and an extract as well, which had got a strong cytotoxic activity. So we selected those compounds for further work. And here is a brief description of some of those compounds. And I will run through two of those which we worked more extensively with it, or three compounds. Now, it was very exciting for me because marine natural products, I had not had a chance to work with marine natural products up until that time. I had only known that there are some drugs which are derived from the marine natural products which I was using in the clinic, but then it gave me an opportunity to actually read more about the MNPs, which are an important source of natural products and therapeutic potential. Obviously, it was mesmerizing to read, to study that 3/4 of the Earth's surface area area of the Earth is covered by water, and we have got more marine products as compared to the terrestrial products. And so there must be something which is very exciting. It was only that it was difficult to get those marine products, and only in 1960s people started extracting drugs from the sea, and then the systematic investigations began in 70s, and then now from 1990 onwards, there are 10,000 or more compounds that have been isolated. And what I know, for example, is that some of those compounds, cytarabine, has been used for the treatment of acute lymphoblastic leukemia and lymphoma and acute myeloid leukemia for last several years, since at least 1980s now. And then two drugs, trabectedin and eribulin, are used for the treatment of breast cancer and ovarian cancer and sarcoma now as well. And those are derived from the marine regions. Here are, one of them is a sponge, other one comes from a tunicate, and this is also from a sponge. And there are the derivatives. For obviously the vast majority of audience today is from chemistry. So you would be very excited to know that there are all these things which you know better than I do. And you can start looking at those things as well. So our question was, we looked at the two most important drugs, the cisplatin and paclitaxel. And we have, I'll share with you the data with cisplatin work that we did. So here are these sensitive cell line, as I said, 2780S, and the resistant cell line, 2780CP, and compared that to normal ovarian cancer over here as well. The paclitaxel work, I think it would be done later, and maybe we can find some collaborators now to see if we can continue doing those work. The first thing that we probably did was not a huge success, We worked on Hibernality, which worked in breast cancer, MCF7 cell line. It was treated, we tried our ovarian cancer, and it was recently accepted for publication. All what we had to say was that it does not work. It does not work because the IC50, even for the sensitive cell lines, is very, very high, and for the resistant, it is nowhere near. So we still had to sort of write a letter to the editor so that people do not spend a lot of time investing on hymenaldicine treating ovarian cancer, at least. Although I would urge a caution over here, it works very well for human adenocarcinoma cell lines here and for lymphocytic leukemia. So if you are interested in other cancers, then yes, hymenaldicine is a good compound. We found it to be effective in MCF7 cell line, but not for the ovarian cancer cell lines. The other compound that we found was Gallic acid. And as you know, it's a polyphenol, which is not only from the identified from the marine organism, but it is abundant in all sorts of flora and fauna, from tea to green tea to grape to berries and etc. And it has got a huge range of of activity, and again in cancers as well. It has been reported, but it had not been reported in ovarian cancer. So we tried to use ovarian cancers. And it just so happens that one of your colleagues working at the ICCBS, who has now moved on to work with us in Oman over the last several years, Dr. Imran, probably he is in the audience as well. He is also a collaborator, and he was very, very kind to accept collaboration. And we worked together, Dr. Imran Hassan, and he told us, he introduced us to this concept of using the congeners. And he said that if you change the position of these oxygen and hydroxyl radicals, you can probably achieve more cytotoxicity. So we use the Gallic acid, which is number 7 over here, and lots of its congeners, and compared that to control. And what we saw on the sensitive and the resistant cell lines, that actually Gallic acid monohydrate was far more effective in terms of cytotoxicity using both the LMR blue assay as well as the CCK8 assay. So we use 2 types of assay just to make sure that we are actually on the right line. And there was a degree of it was highly equal in both CCK8 as well as Gallic acid that we saw cytotoxic activity. So we used, we then went on to combine, which was the basis, the whole idea of doing the experiment, which was to compare to see whether a combination of cisplatin with Gallic acid would indeed be more cytotoxic to the resistant and the sensitive cell lines or not. And indeed, lo and behold, the answer was yes. These are the results from the sensitive cell lines, cisplatin and resistant. These are from the resistant cell lines, cisplatin and Gallic acid. But what we saw over here is something which we were very, very excited, and we continue to be excited about that, is that a combination of Gallic acid and cisplatin, particularly in the resistant cell line, is far more cytotoxic. It reduces the viability down to less than 20% if you combine those two, Gallic acid and cisplatin here. And when we did this nice analysis of the synergy, what we saw also was that the two work very synergistically together as well at different dose concentrations. So we have gone up to there, we have submitted this, we are waiting for the reviewers to see what they say about it. And we also looked at the mechanism of cytotoxicity, and we found obviously that a combination of cisplatin and Gallic acid over there was, it increased the level of caspase-3 more than the controls, and this was significant both in the sensitive cell line as well as the resistant cell line. What you see is that the combination produces an elevated levels of caspase-3 in here compared to the controls. And obviously this was exactly the opposite in the normal cells, which is the host 6-3. So I think this is where we are at the moment. And obviously this requires further work to take it on further from here. I would now, in the last 5 minutes or so, would like to move on to the other compound that we use, which is metformin A1, which is a cyclic pentapeptide, and it is produced by fungi, including the Aspergillus niger over here. There are 3 subtypes, A, B, and C. They are identified from various parts of the sea, and from the Gulf waters, it was metformin A1, which has since been identified and synthesized as well. So we started looking at that. And once again, without showing you the raw data of what are the doses and what are the time and etc., so on and so forth, I would just like to go straight to show you that compared to the untreated cells, cisplatin induces cytotoxicity. Metformin on its alone produces cytotoxicity, but when you combine cisplatin and metformin, it produces a huge cytotoxicity, and that is even at nanomolar, the IC50s at nanomolar levels over here, and you can see the apoptosis here as well as a result of the combination. These are the results from apoptosis. What you see, anything which is in this box, which is the right-hand side box, right-hand upper side box, when you combine the cisplatin with gallic acid, either in the sensitive or the resistant cell lines, you see an increased apoptosis, and this is what we found, and we are very excited to learn about that. The late apoptosis is not a major marker over here, it's the early apoptosis, which you see on the red peak over here. However, we also know that not all the cytotoxicity was induced by apoptosis, because the RNA expression of P53 and BCL2 seen over here, when normalized to the caspase 3 and GAPDH, which is the housekeeping gene, it did not show that significant effect. The protein expression of the several genes are seen over here. So we think that because caspase 9 did not decrease as a result of the combination of gallic acid of metformin A1 with cisplatin, so there may be other mechanisms of cytotoxicity other than apoptosis. Apoptosis is certainly one of them, but the fact that there was also a reduction in actin, we are now working at the moment to see whether actually this combination induces necrosis in addition to apoptosis as well. So that is something which we are doing. So to conclude about the MA1, the metformin A1, it showed promising anti-cancer activity in ovarian cancer cell lines, and a synergistic activity both in the sensitive as well as the resistant cell lines. The synergy was of the highest level, and then we are working to further elucidate the mechanism of action of metformin A1, and I'm very sort of proud that this was published in very recently, actually last month, and you can see Imran over there as well. Where is Imran? Let me show you over here. Imran is over there, and all the collaborators are here as well. Now, today, what we know is that when we started this work in 2013, 14, 15, there were very few drugs, which I showed you. But today in 2021, we, in addition to trabectedin, we have got lurbinectedin, and we have got midostaurin and plitidepsin, which have all been approved for use in various cancers. They come from the tunicates, and then we have got lots of these very exciting compounds over here, which are actually not compounds, which are cytotoxic, but which are used in combination as chemoimmunoconjugates. So what you do is you conjugate those into monoclonal antibodies and then use them. For example, brentuximab I have used already. It is a MMAE conjugated to anti-CD30, which works on the Hodgkin's lymphoma, and this has been in a standard of care now in not only the second line, but a recent randomized trial has shown that it works in first line as well. And then obviously you have got some of the older cytarabine, etc., and there are new compounds which are coming up. So some of the sponges are also very effective as well. So in summary, then I got this opportunity thanks to His Excellency Professor Iqbal Chaudhry for giving me this opportunity to speak to you, to share with you my experiences of what we have been doing across the continuum of the ovarian cancer. I've been involved in looking at the prevalence of BRCA mutations in the population within the Gulf, which is a study which we have just submitted for publication. And found the prevalence to be 17% here. Screening and early detection, I shared with you the data related to E2F5 and FAT4, but there are other data as well. And obviously, for the want of time, I did not share that. But it is all there now and it's all coming up. We are all working on it. Then on treatment, I shared with you some data on frankincense, gallic acid, and metformin. And I think this is a very, very exciting area, particularly with metformin as well as with gallic acid as well. And recently we have submitted an abstract because we looked at the quality of life of patients who survived gynecological cancers and to see what we can do about them. I think in the end, it would not be fair if I did not acknowledge I cannot do all of this work. I did not do all of this work. It was only possible through collaborations. Our 3 PhD students, I think the big credit goes to my colleague, Dr. Yahya Tamimi, Associate Professor in Molecular Biology and Biochemistry, and together with him and lots of other investigators over here who work very closely with me, Shadia, Sergey, Imran, Moza, and Mark, we have looked at or helped 3 people to either get their PhD or they're about to get their PhDs, master's students over there, some of the other colleagues who have either left us or are still working with us, our colleagues who very kindly provided the cell lines, Professor Sang, Professor Saw, and Professor Okamoto. And the work would not have been possible had it not been for 2 major grants. We call it His Majesty grant over here, which is almost to the tune of more than $200,000, which is one over here and one over there. And then some of the other grants as well, which we have been able to get together between me and Dr. Yahya. So with that, I would like to thank you very much. I would like to suggest here or say that in the next couple of weeks, I would be moving on to work in this newly built cancer center called the Sultan Qaboos Comprehensive Cancer Care and Research Center, which is just next door to where I work at the moment. This is on the premises of the university. And I look forward to collaborations with all of yourselves. And I would be happy to take any questions. Thank you very much.
Thank you so much, sir, for your very informative, very exciting lecture. And with this, I think we can move to the question answer session. Sir, if you can have a look in the chat box, we have many questions and lots of appreciation for your talk. So I think, can you open the chat box?
Yes, I did. Thank you very much. And I would now start looking at, it would take time for me to go through all the 92 messages that I have received, that we have received, but let us see if we can. If you would be very kind, please do. That would be great.
So first of all, we have a question from Professor Michael Thurgill. He's asking, does your work at FAT4 suggest that inhibiting the Wnt and the beta-catenin pathways would be beneficial? Interestingly, olaparb also inhibit the TANK kinases, components of the Wnt beta-catenin system.
Thank you very much for that question. And I had the pleasure and the opportunity to attend Professor's talk about a few weeks back. And I learned a lot about the TANK kinases as well. I think these are exciting areas. What we have obviously is the PARP1 inhibition with olaparib. And now Professor has also looked at the TANK kinases as well. And I think the fact that FAT4 is upregulated and can be blocked could be yet another way of treatment of ovarian cancer. So not only a marker of early detection, but also a target for treatment as well.
Thank you very much. So here is also a question, actually two questions, which are sort of asking if there is an effect of hormonal, changes related to somewhat to ovarian cancer. Fluctuation in hormonal imbalances can cause.
Yeah. Thank you for this question. For high-grade epithelial ovarian cancer, no, not a lot, except that, of course, patients who would take estrogens, they would be exposed to get those sorts of cancers. But for the low-grade epithelial ovarian cancers, there is a lot of work actually which is going on to actually suggest that inhibition of the estrogen pathway can actually help to treat the low-grade and the borderline serous ovarian cancers. So it depends on which type of ovarian cancer we're talking about. Yeah.
Right. So here is also another interesting question. From your experience and these results, would you recommend your patients on ovarian chemotherapy to concurrently take herbal medicine product supplements to improve outcomes and quality of life?
Thank you very much for this question. It's a great question. And I know that we are all great believers in conventional medicine and in unconventional medicine as well. Well, when we talk about evidence, we have the evidence. And the evidence at the moment is some sorts of chemotherapy, some types of anti-angiogenesis drugs, some types of PARP inhibitors, now the use of the immune checkpoint inhibitors, which are coming into some forms of ovarian cancer as well. Insofar as the unconventional drugs are concerned, I think the field is wide open. And what we together want to do is to see if we can actually build a database on that, build evidence. I don't see a reason why one of the drugs would not be very active, because we know and we believe that there is treatment for every disease, but it's only a question up until we find the evidence and then use that evidence to treat all ovarian cancers or any other cancer.
Thank you very much. With this, I think we also have some other questions. Does fungi and marine producing drugs cause side effects?
The drugs that we know, trabectedin and eribulin, which are derivatives of the sponge. Eribulin is a sponge derivative, and trabectedin is also a marine derivative, and so is cytarabine. They all produce side effects. The problem with these drugs, the cytotoxic chemotherapy drugs, is that they are not, they do not go onto a particular target. As Professor Threadgill is suggesting, that if you were able to inhibit the FAT4 using a certain compound like tankerases, or like we use, for example, the PARP inhibitors, then the side effect profile would be very different, and the side effects would be minuscule as compared to if you use drugs which basically act either on the DNA intercalation or DNA alkylation, or they inhibit the DNA throughout the body, and then there would be side effects. So there are side effects with trabectedin and eribulin both, and those are managed like any other chemotherapy, basically.
Well, thank you very much. So I think one more quick question. Can you name a biomarker you can use for as soon as possible diagnosis? And number two, can AI help in this diagnosis?
Sorry, what's the second question?
AI, artificial intelligence, help with the diagnosis.
Okay, all right, okay. Thank you very much. The marker which is used at the moment for diagnosis, or people try to use it, is CA125. It doesn't work. And I showed you the UK tox study. It may help to detect ovarian cancer early, but it would not have an impact later on. It is not a very sensitive marker. it is elevated in about 70% of patients with ovarian cancer. And it's not a very specific marker either, because it is elevated in lots of other conditions as well. So at the moment, the serum markers, we don't have any good marker. And I think this is an area for research. Insofar as artificial intelligence is concerned, the answer is yes. I do not know how, because I do not know a lot about artificial intelligence. But if you were to look at for example, the heat map that I showed you, and you could see that hundreds and thousands of genes are upregulated and downregulated, and you put an artificial intelligence engine over there and see what is going where, then certainly, yes, I think that is probably the way in the future, is to look at not one marker, but a combination of thousands of markers, which we, with our usual intelligence, cannot fathom, but with artificial intelligence, we can generate that data. Yeah.
Thank you very much. So I think the last question, at what stage these marine drugs can cure?
These are experimental data. So we have reached a stage where we have used those drugs on the cell lines. Obviously the next step is to go and try them out in the animal models as well. If one of you would be, or some of you would like to collaborate, that would be a wonderful opportunity to collaborate. And then they go on to clinical trials. So at the moment, the drugs that I've shown you on the slide, I think third or fourth from the last, are the drugs which have come into clinical practice. So those are being used for ovarian cancer. Trabectedin and eribulin both are being used, but for these compounds, it's some time to go.
Right. With this, thank you very much, sir, for a very comprehensive and interesting talk. And I would request Dr. Hina Siddiqui if she has something to say about this.
Thank you so much, Hazima. Nothing but to say thanks, a big thank you to Professor Ikram. Such a wonderful, informative talk, and certainly one of the most popular lecture. I saw house full and lots of appreciation and questions. These are reflecting the the understanding and the way you presented your lecture was wonderful. Thank you so much, and we look forward to more and more interactive lecture in future as well. Thank you so much, sir.
I thank you very much, and I thank His Excellency Professor Iqbal very much for this opportunity, the ICCBS and the Comstech. It was indeed a pleasure and a privilege for me to be here, and I look forward to collaboration, inshallah. Thank you.
Thank you very much, Professor Ikram. And I would also like to thank our participants who joined us today and for keeping this session an interesting and interactive one. Thank you very much. Hope to see you all in some other event, inshallah.
Thank you.
Bye-bye.
Assalamu alaikum.